Welcome to Dhanu's Academy. Today's video is about amino acids and proteins. Contents of today's video: general structure of amino acids as amphoteric molecules, optical properties and specific rotation of amino acids. Amino acids are compounds containing carbon, hydrogen, oxygen and nitrogen. They serve as monomers, building blocks of proteins and composed of an amino group, a carboxyl group, a hydrogen atom and a distinctive side chain, all bonded to a carbon atom, the alpha carbon. In an alpha amino acid, the amino and the carboxylate groups are attached to the same carbon atom, which is called the alpha carbon. The various alpha amino acids differ with respect to the side chains or the R group attached to the alpha carbon. The general structure of amino acid is depicted in the slide. This structure is common to all except one of the alpha amino acids, that is proline, which is an exception. The R group or side chain attached to the alpha carbon is different in each amino acid. In the simplest case, the R group is a hydrogen atom and the amino acid is called as glycine. In alpha amino acids, both the amino group and the carboxyl group are attached to the same carbon atom. However, many naturally occurring amino acids which are not found in protein have structure that differ from the alpha amino acids. In these compounds, the amino group is attached to a carbon atom other than the alpha carbon atom, and they are called beta gamma amino acids depending upon the location of the carbon atom to which the amino group is attached. The structure of amino acid lysine is depicted in the slide. Amino acids act as acids and bases. When an amino acid is dissolved in water, it exists in solution as the dipolar ion or Zwitterion. A Zwitterion can act as either an acid, proton donor, or a base, proton acceptor. Hence, an amino acid is an amphoteric molecule. At high concentrations of hydrogen ions, that is low pH, the carboxyl group accepts a proton and becomes uncharged, so that the overall charge on the molecule is said to be positive. Similarly, at low concentration of hydrogen ion, that is high pH, the amino group loses its proton and becomes uncharged. Thus, the overall charge on the molecule is said to be negative. The acid-base behavior of an amino acid in solution at low pH, the positively charged species predominates. As the pH increases, the electrically neutral Zwitterion becomes predominant. At higher pH, the negatively charged species predominates. All amino acids except glycine are said to be optically active. That is, they rotate the plane of plane polarized light. Optically active molecules contain chiral carbon. A tetrahedral carbon atom with 4 different constituents are said to be chiral. All amino acids except glycine have chiral carbon and hence they are optically active. An optically active compound can rotate the plane of polarized light either clockwise or counterclockwise. Optically active compounds that rotate the plane of polarized light clockwise are said to be DEXTROROTATORY. By convection, this direction is designated by a plus sign. Optically active compounds that rotate the plane of polarized light counterclockwise are said to be LEVOROTATORY. This is designated by a minus sign. The plus and minus forms have also been termed as D and N respectively. When plane polarized light is passed through a solution that contains an optically active compound, there is net rotation of the plane polarized light. The light is rotated either clockwise or counterclockwise by an angle that depends on the molecular structure and concentration of the compound, the path length and wavelength of light. Optical activity is measured by polarimeter. The magnitude of the optical activity is measured as an angle of optical rotation. A quantitative measure of the optical activity of the molecule is known as optical rotation. Optical rotation of an optically active compound depend on the following: concentration of the compound, light path length, the wavelength of the polarized light, and the temperature. Hence, experimentally measured optical rotation is always converted to and expressed as specific rotation. The optical rotation of a solution at a given temperature and wavelength is given by the following equation. Specific rotation is equal to degree of rotation divided by L into C, where degree of rotation is the observed rotation in degrees, the specific rotation, which is the specific rotation of compound at temperature T in degrees Celsius, and wavelength lambda in nanometer. If the wavelength of the light used is 589 nanometer, the symbol D is used in the specific rotation. C is the concentration of the solution in gram per ml. L is the light path length in decimeter. Now, we will be solving a problem which is based on the specific rotation. A solution of L-LEUCINE 3 gram per 50 ml of 6NHCl had an observed rotation of plus 1.81 degree in a 20 centimeter polarimeter tube. Calculate the specific rotation of L-LEUCINE in 6NHCl. As we know, we have written the formula. Next, we will have to take all the data given in the question. The degree of rotation is given, the length is given, concentration is given, but L is in centimeter, which has to be converted into decimeter. Concentration is given per 50 ml, which has to be converted into per ml. Then just substitute the degree of rotation L and C in the formula. Then after solving, we will get the specific rotation as plus 15.1 degree. Thank you. If you like the contents of our video, please like, share and subscribe to our YouTube channel.